Hello Biology 100 students, welcome to your first lecture on the circulatory system. Today we're going to talk about blood, which is an amazing connective tissue which helps to carry nutrients throughout the body and also provide the body cells with oxygen and immune defense. Alright, so there's three basic components to the circulatory system. First we have the heart. The heart is the pump that circulates the blood throughout the body. We have the blood vessels, which are the tubing or the pipes that get that blood where it needs to go. And of course we have the blood. That is the tissue that's carrying the oxygen, transporting waste products, transporting hormones, etc. Alright, so there's several important functions of blood that we'll talk about today. Firstly, it helps to transport oxygen to the cells. That's the job of our red blood cells or erythrocytes. It also helps to transport nutrients from the digestive system to the body cells. That's happening within the blood plasma. It's also important for transporting hormones from their source tissues to their target tissues. For example, it transports insulin from the pancreas to the target tissues throughout the body. And of course, it transports waste products as well, such as things like carbon dioxide, urea, etc. It's also very important in distributing body heat. I mean, think about what happens to your skin when you're working out really hard. You begin to overheat and you'll notice your skin gets really red. And that's because the blood vessels in your skin dilate, bringing the blood closer to the surface, and that helps to cool off the body. And last but not least, the circulatory system in the blood is important for pH buffering, maintaining the ideal balance of hydrogen ions in the blood so that we can have homeostasis. All right, now let's talk about the physical and chemical characteristics of blood. Firstly, blood is thicker than water, as you probably know. You've probably cut yourself at some time. It also has a higher temperature than body temperature. It's around 100.4 degrees Fahrenheit, whereas body temperature is around 98.6. It has a pH of 7.4, so it's slightly alkaline, and the optimal range for pH is 7.35 to 7.45. If it becomes too basic, you have a problem. Too acidic, you have a problem there as well. Now, blood is actually about 8% of our total body weight. So a lot of our body weight is actually blood. And it depends on whether you're male or female. So males have a little bit more, around 5 to 6 liters. Females have around 4 to 5 liters, depending on body size. And there's a hormonal native feedback loop that controls the number of blood cells and also helps to control blood volume. Okay, remember back to our lecture on tissues, and we said that blood was a specialized connective tissue. And connective tissues consist of some cells, but they also have a lot of extracellular matrix. So the solid component here is mainly cells, right? We're talking about red blood cells, white blood cells. And the liquid component up there is going to be the matrix. And the matrix here consists of plasma. And plasma is basically water with a lot of proteins in there as well. Now, of the blood cells or formed elements, about 99% of those are going to be red blood cells. So a major function of blood, of course, is carrying oxygen. Less than 1% are white blood cells and platelets. Now, white blood cells will be important for immune defense, and platelets will be important for blood clotting. Okay, you've probably gone to the doctor's office sometime and had some blood drawn and maybe even saw them spin it down. You can see this liquid component right here, the plasma, the solid component down here consisting of the formed elements or cells. What you might not notice is this part down here is the red blood cells, and then we have something called the buffy coat, which is a layer of white blood cells and platelets. So the thickness of this layer, ironically, can actually tell us if we have an infection, because when we have an infection, oftentimes we have an increase in the numbers of white blood cells, and that buffy coat will be a little bit thicker. All right, but talking about plasma, plasma is the stuff that's up here. So plasma consists mainly of water, but it also consists of plasma proteins such as albumin, globulins, and fibrinogen. Let's talk about albumin. Albumin is a protein you might have had for breakfast. That's because it's found in eggs or egg whites. Now in our bloodstream, albumin helps to maintain our osmotic balance. On the other hand, globulins or immunoglobulins help to defend us against infections. You've heard of antibodies. Antibodies is another name for immunoglobulins. And finally, the last protein here is something called fibrinogen. Fibrinogen is a soluble protein which helps with blood clotting. So if you get a cut in your skin or something like that, fibrinogen is a very important part of the clotting cascade, which helps to stop that bleeding, and that process is called hemostasis. Okay, let's go on to talk first about the formed elements. Now, we're calling them formed elements rather than cells because one of them is not a cell. Now, red blood cells, white blood cells, those are cells, but platelets are not cells. Platelets are actually cell fragments. So we have our red blood cells, otherwise known as erythrocytes. Erythro here means red. And then we have our white blood cells known as leukocytes, and those are divided into granular and agranular, as we're going to talk about later. Now, platelets are called cell fragments, or they are cell fragments, and they're derived from something called a megakaryocyte, which hopefully is down there somewhere. Now, the important part to realize is that all these blood cells are made in a red bone marrow. The red bone marrow was located inside the long bones and the flat bones of the hips, so this is where we're producing our red and white blood cells. Okay, let's go on to our first formed element, and that's going to be our erythrocytes. Erythrocytes are our red blood cells, and remember their number one function was to transport oxygen from the lungs to the body tissues. Now, red blood cells are really weird cells. They're very, very small, around 7 microns or 8 microns. They're biconcave, they look like Cheerios to me, and they don't have any organelles. They don't have a nucleus, they don't have mitochondria or anything like that. And the reason is that helps to free up space so we can have more room for the hemoglobin. And hemoglobin is this pigment that helps to bind to oxygen and transport it throughout the body. But as a result of not having a nucleus or organelles, they don't live very long. They live about 120 days, so they need to be replaced all the time. Okay, so interesting facts about our red blood cells, or erythrocytes. One red blood cell contains about 250 hemoglobin molecules. And because each hemoglobin molecule can carry 4 oxygen molecules, one red blood cell can carry up to 1.2 billion oxygen molecules. That's pretty cool. Now, the interesting thing about red blood cells and hemoglobin is they change color depending on oxygenation. If you look at arterial blood that's very well oxygenated, it tends to be bright red. On the other hand, venous blood, which is depopulated of oxygen, tends to be purple or blue. And as I was saying earlier, our red blood cells only live about 120 days, so we're making new red blood cells all the time. On average, we're making about 2 million red blood cells every second. All right, like everything else in the body, the circulatory system and the numbers of red blood cells is regulated through homeostasis. Now, the important organ in here is the kidney, because the kidney is monitoring blood oxygen levels. And so if blood oxygen levels begin to fall, what will happen is the kidney will secrete a hormone called erythropoietin. Erythropoietin acts on our red bone marrow, and it tells our red bone marrow to make more red blood cells. As a result, we make more red blood cells, and as a result, our blood oxygen levels increase, and that results in a decrease in erythropoietin. So this is a classic negative feedback loop. Now, while we're talking about red blood cell numbers, I need to talk about some of these strategies that athletes use to increase their hematocrit, or numbers of red blood cells. So a lot of athletes, really endurance athletes, may train for a long time at high elevations. And they do this because higher elevations have a less partial pressure of oxygen. So they're kind of hypoxic, lower than normal oxygen levels. And by training at those areas, one, they build their endurance, but two, they may build their hematocrit if they're up there for a long time. And that's because as they're up there experiencing hypoxia, the kidney begins to ramp up production of that erythropoietin, and erythropoietin causes us to produce more red blood cells. And theoretically, more red blood cells should give us better endurance when we're back down at sea level and competing again. Okay, so now we're going to talk about our leukocytes, which are our white blood cells, and their function is primarily immune defense and warding off infections. So let's talk about their structure. First of all, you can see they're much larger than the red blood cells. The red blood cells are these little pink dots in here, and then the white blood cells are much larger, and they all have a nucleus, right? So we can identify them very readily. So they're larger, they are comparatively rare, they have a nucleus, they do not contain hemoglobin, they don't carry oxygen. And there's two different types of leukocytes, granular and agranular. The granular ones have granules, and the agranular ones do not. So they are made in the red bone marrow just the same as the red blood cells, but some of them will actually go to mature in the thymus, and these are our T cells we'll talk about in a little bit. Now, it's important to realize that some white blood cells can actually leave the bloodstream and enter the tissues. And this is important because a lot of times an infection can start out in the tissues. Think about stepping on a rusty nail or something like that. If it had some bacteria on it, that bacteria is going to enter the dermis of your foot, and we need those white blood cells to be able to go out of the blood vessels and attack the bacteria within the tissues itself. Now, the process through which a white blood cell moves out of the bloodstream into the tissues is called diapedesis. So diapedesis is a pretty complex process involving chemical signals that tell the white blood cell to emarginate on the wall of the capillary bed and then squeeze out in between the walls of that blood vessel to enter the tissues. But suffice it to say, if we have bacterial proteins or anything out here, that's going to attract these white blood cells and cause them to go through this process of diapedesis. All right, our first group of white blood cells are called granulocytes. And as the name implies, these have granules in them. There's 3 different types of granulocytes: neutrophils, eosinophils, and basophils. And they're named based on how the granules stain with this sort of right stain that we use to stain blood cells. So neutrophils up top, their granules are there, but they stain neutrally with the dye, so we don't really see them. But what we do notice about a neutrophil is it has this segmented nucleus. Now, neutrophils are important first responders. They're the ones that deal with bacterial infections. So you stepped on that rusty nail, there's some bacteria on it. Within hours, they're going to be moving out of your blood vessels into your tissues and gobbling up any type of bacteria that are present. Now, they kill the bacteria through the process of phagocytosis. Phagocytosis is literally gobbling them up, and then we have lysosomes and things like that that bind with the bacteria and actually destroy it. So the second type of granulocyte is something called an eosinophil. Now, we call them eosinophils because they stain very brightly with this orange to pink stain called eosin. So they have these nice pink to red granules, and they have a nucleus that looks like an old telephone or a set of headphones. Now, eosinophils don't really deal with bacteria, but they do deal with large parasitic organisms, for example, worms or something like that. And lastly, our final granulocyte is something called a basophil. Basophils are very, very, very rare. We actually have to look through a blood smear for quite a long time to find them. And basophils don't actually attack parasites or anything like that, but they do secrete something called histamine. Now, histamine you've heard of before. You've probably taken an antihistamine. You take an antihistamine to relieve symptoms that involve congestion and things like that. Histamine, on the other hand, is the thing that causes the congestion. So histamine causes inflammation, and it causes blood vessels to become leakier. And this is an important part of the sort of cellular repair process, because when we have an injury, histamine helps to make the blood vessels more leaky and allows the white blood cells to move out and provides nutrients for the tissues that are being repaired. All right, now we're going to move on to our agranulocytes. So these things don't have granules. And our first agranulocyte is something called a monocyte. And monocytes are huge, very, very large compared to a red blood cell. Can be 2 or 3 times the size of a red blood cell. And they have a U-shaped nucleus that looks like a horseshoe a little bit, or looks like a kidney bean. And these guys differentiate into something called macrophages. Macro here means big, and phage means eating. So they gobble up bacteria, cellular debris, and all kinds of things. They typically arrive later than the neutrophils, but they also do a lot more scavenging and eating up of bacteria than neutrophils do. Okay, our next agranulocyte is something called a lymphocyte. Now, lymphocytes are a lot smaller than monocytes on average. They tend to have a big nucleus in comparison to the cytoplasm that's out there. So lymphocytes are sort of like your cellular hitmen. They're not normally out to kill just any type of bacteria or virus, but they have specific immune targets. So they have a specific target. They may not bother with this bacteria or that bacteria, but they're going to specifically target one type of bacteria or one type of infected cell. So we have 2 different types, B cells and T cells. And these are named for where these cells undergo a process called immunocompetence. Immunocompetence is sort of like the SEAL Team 6 school for SEALs. It's where they learn what their targets are, and they really go through this rough process of learning how to be good B and T cells. So B cells undergo their schooling in the red bone marrow, and T cells undergo their schooling in the thymus, which is a gland surrounding the heart. Now, B cells don't directly attack a pathogen. Instead, they secrete something called antibodies or immunoglobulins, which then might attack a particular pathogen, whereas T cells directly attack infected body cells. All right, our next blood component are platelets. Now, platelets really aren't cells. They're actually parts of cells that were formed through fractionation of something called a megakaryocyte. And the purpose of platelets is to stop blood if we have a broken blood vessel. So they help to do hemostasis. So if we're bleeding because we've torn a blood vessel or ripped a blood vessel, the platelets along with our fibrin or fibrinogen will help to stop that bleeding. So here you can see in this picture an SEM micrograph. There's some red blood cells. Remember, those are pretty small. Platelets are even smaller. And here they're just kind of floating around in the blood like sort of benign Rice Krispies. But as soon as that blood vessel is torn or something like that, these platelets become activated. They sprout these little appendages called pseudopodia, and they go through a positive feedback loop where they activate other platelets. And this helps to start the process of hemostasis and stop the blood flowing so we don't die of blood loss. So hemostasis, as I was saying, is stoppage of bleeding in a quick and localized fashion when blood vessels are torn or damaged. And it prevents hemorrhage or loss of large amounts of blood. And there's 3 main steps we're going to talk about today. 1 is vascular spasm, 2 is platelet plug formation, and 3 is blood clotting. Okay, let's start with vascular spasm. What causes it? So first of all, you stepped on that rusty nail and then blood's coming out of your foot. Just the damage to that blood vessel is going to cause a reflex in there where the smooth muscle in that blood vessel will start to contract. Now that smooth muscle will reduce the diameter of the blood vessel and hopefully reduce the blood flow so we don't die immediately. Now, if this happens in a small blood vessel, like in your face when you're shaving, it works pretty well. If you hack off an arm, this vascular spasm isn't going to be very effective because the blood pressure in your arm is quite a bit higher and we have a larger blood vessel to start with. Okay, the next step in our hemostasis process is something called platelet plug formation, and this has a couple sub-steps. So firstly, let's talk about our platelets. Normally they're floating around in the blood plasma and they're inactivated. They don't have any pseudopodia. So normally as they're bouncing against the walls of the blood vessel, they're just encountering endothelium, and that's an epithelial-like tissue lining the blood vessels. But when the blood vessels are torn, that's going to expose the underlying connective tissue layer, which normally the platelets would not be encountering. So as soon as they bounce against some collagen or something like that, they are going to become activated. And when they become activated, their little pseudopodia come out and they grab a hold of other platelets as well and activate them as well. And we also secrete a lot of chemicals, which then are going to get our fibrin or fibrinogen involved. Remember, fibrinogen was sort of like this soluble protein which is floating around in the blood plasma, and normally it's not doing anything, except when the platelets become activated, that fibrinogen gets converted into something called fibrin, and now we end up with a platelet plug that is wound up with lots and lots of fibrin threads, and these fibrin threads help to solidify the plug and stop the leak. Okay, so normally hemostasis is a good process that helps us to heal or seal broken blood vessels. However, sometimes clotting can happen even though we don't have a break in a blood vessel, and this can be dangerous. This process here is called thrombosis. So thrombus is a clot in an unbroken blood vessel. And what are the causes? Well, one thing, we could have a rough lining in there that's initiating our activation of the platelets. The other thing is we could have really sluggish blood flow, and that can cause clotting as well. Think about that 6 or 7 hour plane ride you might have over from the mainland to Hawaii, and as you're sitting there, if you're not moving around particularly, if you're older, just sitting there can cause clotting to start to form in some of these deep veins. Now that's dangerous enough in itself because it can deny blood flow to the downstream areas of that blood vessel. But that clot can also break loose and become something called an embolus. So if we go from a thrombus to an embolus, this is really dangerous because this floating blood clot is going to float around the bloodstream until it finds somewhere to stick. And oftentimes that can be in the brain or the lungs, and it can be very, very dangerous or even deadly. So to reduce the chance of embolism or thrombosis, some people take low-dose aspirin, and that tends to reduce the chances of blood clots. Okay, before we wrap up this lecture, I want to talk about blood types. Now, some of you might already know your blood types, and if you don't, you can give blood at the blood bank, and they'll send you a little card back in the mail and tell you your blood type. Now, it can be beneficial to know your blood type because that can tell you who you can potentially receive blood from. So there are 4 different blood types we'll talk about today: A, B, AB, and O. And as many of you know, there's also a positive and negative, but we're not going to spend much time on that. Now, in order to understand how blood types work, we first have to understand what antigens are. An antigen, theoretically, is anything that can initiate an immune response. In this sense, an antigen is a surface marker or glycoprotein that's on the surface of our red blood cells. So this allows our body to tell the difference between our own cells and cells that would be pathogenic or invading cells. So that way we can attack the pathogen cells, like the bacteria, but we're not going to attack our own cells. Now, most cells, including red blood cells, have special antigens on their surfaces. On the other hand, your body makes antibodies. Antibodies attack antigens which are foreign to the body. So we make antibodies to the antigens we don't have so that if those antigens are encountered in our body, we will attack them because those must be foreign cells. So the big picture here is that your white blood cells make antibodies to the antigens you do not have. If you made antibodies to the antigens you did have, you would always be attacking your own cells, and that would be an autoimmune reaction. All right, so the big picture here is that your blood type is based on the presence or absence of special glycoprotein antigens on the surface of your red blood cells. And the major antigens we're going to talk about today are the A and B antigen. Remember, the immune system will make antibodies for all the non-self antigens. In other words, it makes antibodies to the antigens you do not have. So if you have type A blood, that means you have type A antigens, and you will make type B antibodies. You wouldn't make type A antibodies because that would attack your own cells, and that would cause an autoimmune reaction. It's important to realize you can only receive blood from donors whose blood does not contain antibodies against your own cells. All right, so let's look at an example. This person has type A blood, and that means that their red blood cells, erythrocytes, have type A antigens on the surface. Now, really, the antigens don't look like giant A's. That's just there for illustrative purposes. Now, remember the rule. We're going to make antibodies to all the antigens we do not have. Well, how many antigens are there? In this case, there's just 2, A or B. So which antigen do we not have? Well, we don't have B. In that case, we have anti-B antibodies floating around out in our blood plasma. Now, these don't interact with our cells because we don't have B antigens. But if somebody gave us B blood, our antibodies would then attack their antigens and destroy those red blood cells. All right, now let's take a look at somebody with type B blood. So same red blood cell, but now we have B antigens on the cell surface. Remember the rule, we make antibodies to all the antigens we do not have. Which antigen do we not have? Well, we don't have A. So out here, we would have anti-A antibodies. So I could not receive a blood transfusion from somebody with type A blood because my antibodies would attack that blood. Now let's look at somebody with type AB blood. That's what I have. So if you have type AB blood, your red blood cells have both the A antigen and the B antigen on their surface. What antibodies do we have? Well, remember the rule, we only make antibodies to the antigens we do not have. Well, what antigens do we not have? We have both antigens, A and B, therefore there's no antibodies out here. Now the good thing about somebody with AB blood, because they don't have any antibodies in their serum right there, they can get a blood transfusion from somebody with type A, type B, or type AB blood. Okay, now let's take a look at somebody with type O blood. It's important to realize there's no O antigen. I want you to think of O here meaning 0. There are 0 antigens on the surface of the cell. So no A antigens, no B antigens, etc. Now which antibodies will we produce? Well, remember, we produce antibodies to all the antigens we do not have. We have neither A nor B antigens, so we make both anti-A and anti-B antibodies. What does this mean for somebody with type O blood? Well, they can't get A blood because the A antibody would attack it. They can't get B blood because the B antibody would attack it. And they can't get AB blood because both antibodies would attack it. So somebody with type O blood has to get blood from somebody else that's an O. Okay, last but not least, I'm just going to say something about Rh groups. If you've gotten your blood typed, or if you know your blood type, you probably know whether you're negative or positive. So the negative or positive here indicates the presence or absence of something called the Rh antigen. And this was named for the rhesus monkey, not the rhesus pieces. And so people that have the Rh antigen are said to be Rh positive, and people that don't have the Rh antigen are said to be Rh negative. So the important point here is that people that are negative can only receive from other people that are negative. People that are positive can get blood from people that are positive or are negative. Now, not only is it important to know the Rh factor if you're getting blood or giving blood, but it's also important if you're having children. If, for example, you're an Rh negative mother and you've already had one child, if that child ended up being Rh positive because of genetics, you could then start raising antibodies against the Rh antigen. And if you have a second Rh positive child, you could actually have an immune reaction against your own fetus. So it's important to know whether or not you are Rh negative or Rh positive. And if you're an Rh negative mother, they're probably going to give you a RhoGAM shot that keeps you from raising antibodies against your own child. All right, you've reached the end of the lecture on blood. This is the first part of the circulatory system. As always, if you have any questions, go back and review that in the lecture. If you have any questions that can't be answered that way, send me an email or a text. Thanks.